Welcome to the Molecular Devices Automated Electrophysiology webinar series. We're going to go ahead and get started now. I'm Jim Costantin. I'm the host of the webinar. Xin Zhang will also be helping me today, hosting the webinar. We have three speakers today. Sorry, technical problem. Our three speakers today are Juha Kamanen from Pfizer Nucentis in Cambridge. We have two speakers from Bristol Myers in Connecticut, Adam Hendrickson and Jim Harrington. Juha will be talking about his experiences with the IonFlux HT system. Jim Harrington and Adam Hendrickson will be talking about their experiences with the Ironworks Barracuda system. And then we have joining us today also Ali Yeehaw from Fluxion, talking about the IonFlux instrument. As I mentioned, I'll be hosting along with Xin. I wanted to give you a brief introduction about some of the tools that we make for ion channel drug discovery. So we're looking at the drug discovery funnel here. Basically, at the top of the funnel, we have the flipper, which is a fluorescent plate reading instrument for high throughput, low cost screening. Going down the funnel, we have the Ironworks Barracuda system, and as well as the Patch Express system. You can see we've put arrows in where they approximately fit into the funnel. At the bottom of the funnel, we have the conventional electrophysiology equipment, or axon electrophysiology, and then we also have the IonFlux system, where I didn't actually add an arrow because we actually are finding that the two systems fit quite nicely all along the funnel. So there's the IonFlux HT higher throughput system and the IonFlux 16 system. After the webinar, you can view this as a recorded webinar or any of our other previously recorded webinars by navigating to our webpage and going to either the IonFlux or the Ironworks Barracuda webpage, and then clicking on the window to view the webinars. And with that, we'll go ahead and get started with our first speaker, who's Juha from Nucentis, and I will go ahead and pass you the ball here, Juha, if that's okay, if you're ready.
Yep. Can you hear me all right?
Yep, I can hear you just fine. So I'm going to go ahead and pass it over to you.
Yep.
I'll let you know when I can see your slides.
Okay.
So Juha, have you shared the application?
Yeah, just give me a second. Once I can, there we go. Now I can access. Okay.
Okay, yeah, I can see your slides.
Cool. All right, so I'll just get started.
Great.
The slides are more or less the same that I showed earlier, but I think there's some valuable data from across different types of ion channels. Just a brief snapshot of what we've been doing. There's some screening data which I can't show just yet, and also there will be a poster at Vancouver Ion Channel Retreat together with gene data about some recent stuff that we've done, specifically related to analysis. But I'll show data, and I think Ali's going to mention a few things in detail about the protocols and different opportunities later on. So the primary target that we initially looked at the IonFlux for was GABA-A, and the reasons were that we wanted something where we'd be able to run ligand-gated channels with very good control over the liquid applications. And that's what we really like about the IonFlux. We're able to set up pretty complex liquid application protocols, and you know, the responses are very quick. The fact that you can apply multiple different liquids to the same cells allows you to control your responses or normalize your responses to multiple different controls. So in the experiments that I'll show you next, we've applied reference GABA multiple times. We've then applied diazepam as a positive control for allosteric modulators. Then we've added a test PAM. We've then washed that out, reapplied diazepam to check what sort of enhancement we get. And then finally we applied 100 micromolar GABA in order to check where we are on the GABA EC50 curve. So what do I mean by setting up complex application protocols? In the screen here you can see, let me just put my pointer on constantly. What we're applying here from the 8 different application wells, first of all reference GABA, then we're applying, in channel 7 we've got the vehicle control with DMSO, then we've got reference GABA, and then we've got applications from different compound channels for different allosteric modulators, and then we've got the 100 micromolar GABA applied towards the end of the experiment. And in this magnified view of the application protocol, the point that I want to make is that you can overlap and overlay liquid applications. So what's happening here is that from channel 7 I'm applying the vehicle, and from channel 6 I'm applying a compound, and the 2 liquid applications overlap so that when the vehicle application finishes you get a pretty much instantaneous liquid exchange. So that's done for both the concentration of compound here as well as down here. And hopefully what you can see is the end result is instantaneous changes and very fast responses. So this is for GABA A using an in-house cell line with alpha-2 subunit. We're applying an EC20 of GABA and then different positive allosteric modulators in an open channel assay format. And what that means is that we first of all apply GABA to open the channels, and then we apply different compounds or DMSO, and then measure the enhancement of the current by these positive allosteric modulators. So what I hope you can see is that we can get very stable responses with GABA, and then very fast responses to different positive allosteric modulators. The graph on the left is just a single concentration of the positive allosteric modulators. The graph on the right shows a concentration response to the positive allosteric modulators. So you can see in green we have diazepam with increasing concentrations being applied and overlapped, and then we can measure the enhancement at different time points here. The way we like to analyze ion flux data is using the gene data screener or nowadays analyzer application. The reason being that it's possible to analyze multiple plates of data at once, and it's also a pretty quick method. You can set up your own calculation cursors, multiple at the same time. And what I'm showing here is the experiment that I described earlier. So we have multiple reference GABA applications. Then we have diazepam applied here. We're washing that out, and then we're applying diazepam again in a concentration response, and then washing that out back to baseline, and then finally applying 100 micromolar GABA to check where we are in the EC50 curve. So you can see that you can set up pretty complicated experiments, get a lot of data out of your experiments, and you're able to normalize responses to internal controls. I've just highlighted here the kinds of calculation cursors that we can set up. So here we're measuring the level of current from baseline, and then the same thing in the concentration response. We're measuring the level of current in this particular zone, this concentration, versus baseline. And then all of these are normalized back against the level of current from the GABA. Okay, so what does that look like when you plot the data? On the left-hand side, I've shown the data that is generated from the single concentration protocol. So we're looking at DMSO diazepam, LA38, and a couple of other known positive allosteric modulators. These responses have been normalized to the first diazepam response within the same cell, and therefore, because we're getting some desensitization and an imperfect washout, perhaps, in case of some compounds, diazepam comes down to about 80% of the initial diazepam response. But this is something that's pretty typical and would happen on manual patch as well. And on the right-hand side, I then have EC50s plotted for the positive allosteric modulators from the experiments that I showed earlier. And we're getting EC50 values which are in pretty good agreement with published data. And all of these experiments can be run very quickly. That's the other benefit of the system. The application protocol that I just showed you is a complete experiment, and you can test 32 compounds in an ion flux HT plate in a protocol which lasts a number of minutes. So it's very, very quick. Another ligand-gated target, and this was something that we ran as a bit of a demo for someone where we'd never used these cells before. Essentially, we received them. They're a millipore, ready-to-assay cell line, HEC, nicotinic acetylcholine receptor alpha 1. Essentially just thawed the cells in an external buffer, put them straight into the ion flux plates, and applied a few different agonists and ligands. So I wanted to show you here the speed of the response for nicotinic alpha 1. It's pretty rapid. If you look at the time scale on the bottom, this is in milliseconds, so we're getting very fast responses. And this is the protocol, again, has been set up to overlap the liquids, so you get instantaneous liquid exchange. This is the current versus time plots from that experiment, just showing what the currents look like with increasing concentrations of agonists. And essentially that's then been, we've taken the current values from the peaks and plotted those as EC50s for either acetylcholine or epipatidine, which is another known agonist. And basically the EC50 values that we've got from this one experiment, never run before with these cells or compounds, and we get values which are in fairly good agreement with published values again. And I guess the message here is the ease of use of the instrument. You can take a cell line that you haven't used before, set up an application protocol, put the cells on, and you should get pretty good data straight off the bat. Voltage-gated targets, so we've been using the millipore, I should say urofins here, obviously. Old habits die hard. And we've been using the urofins HEC, human NaV1.8 plus beta-1 cells, and been running those. So on the right-hand side, I'm just showing some example current sweeps from an IV experiment. And then on the right-hand side, showing the derived values in a plot. What I would like to point out in the right-hand graph is that because we're using the ensemble plates here, measuring currents from up to 20 cells, for a cell line which doesn't routinely express a large number of the channels, we're able to get very good combined current amplitudes, and therefore a good assay window. And we're actually running some screening right now on the ion flux for voltage-gated targets as well. And just to show you the comparison, here's the data from the urofins, or millipore brochure for this cell line, and our IV is in fairly good agreement with the published values. There's a little bit of a shift in the peak and the reversal, but the buffer solutions were ever so slightly different. And we didn't have any compensation switched on here. So I'm quite happy with that. Same sort of experiment this time, steady state inactivation. Again, our V-hat value is in good agreement with value from the Eurofins brochure. Slope is similar as well, so I'm pretty happy with that. And again, what you're able to do on the ion flux, thanks to the liquid application, is you can put on different concentrations of the same compound, or potentially different compounds in a more screening-like mode, with wash-offs in between. So that's what I'm showing here. So from channel 8 in this case, I'm applying the vehicle controller, DMSO, and from channel 7 to 1, I'm applying increasing concentrations of the test compound. So this is a 7-point concentration response experiment. And again, we're overlapping the liquid applications so that we get instantaneous change of the liquid. And in our case, this change takes place in between the measured sweeps. Therefore, the first sweep of your compound application should already show the block beginning. You don't have to do it this way, but we found it to be quite useful in order to speed up experiments a little bit. Here's an IT plot of that same experiment. So you can see in brown, we've got our DMSO control showing you that the currents are pretty stable over a long period of time. So the time scale here is in seconds, so you can see that our total experiment time here is about 40 to 45 minutes. So the cells last really quite nicely on the ion flux instrument. What I'm also showing is compound responses to those increasing concentrations. And one thing that I do want to point out is that, because of the biology and partly the compound physicochemical properties, some of these don't necessarily wash out completely. In this particular experiment, we've used a fully inactivating protocol throughout the whole experiment. So I can highlight 2 points here. So if you're comparing, for instance, the blue trace, which is a standard local anesthetic reference compound, you can see that after the compound blocks some of the current, once you wash it out, it comes back to baseline very nicely. However, if you look at the compound in purple, you can see that upon wash-off, it comes off very slowly. And when we then plot the IC50s for these compounds, I'm highlighting a few compounds here which don't come off completely. There's the Abbott reference compound, and then a couple of our own, where the wash-off is incomplete, and therefore the percent inhibition per concentration is misleading. It's underestimated because the baseline that we're calculating from isn't quite right. Now there is a way that with the latest version that I think Ollie's going to talk about, latest software version, you can stack your voltage protocols. So you could introduce perhaps a resting state voltage protocol for the wash-off, and then go back to your inactivated state for compound application. So something that's on the list of things to do. But I think overall, and I hope you agree, the data quality is pretty good. So I'm showing here averages and standard deviation from 4 experiments. And then correlating to the Patch Express, it's the same compounds as above, and I hope you can agree with me that the correlation overall is pretty good. The potency of some of the compounds which don't completely wash out, for instance, the Abbott compound, they are somewhat off the line. And also the other aspect is that the experiment here on the Patch Express was using the steady state detection for compounds, so the compounds were applied for a longer period of time, whereas we have a fixed limit to the time that each concentration is applied, and therefore we might underestimate the potency. But I would say the correlation overall is pretty good. And just as a sort of final point before handing over. How have we incorporated the ion flux into our operation? As a background, we've got a multitude of different automated EFIS systems in the lab, and we're responsible for generating both the lead optimization, but also increasingly up to HDS scale data. So how do you then minimize time and maximize throughput? For example, we're able to, thanks to the SBS standard plate layout for the ion flux, we're able to dispense compounds directly from a DMSO stock plate into the ion flux plate consumable on, for example, the mosquito, and then simply dilute the plates with external buffer afterwards. Or alternatively, you can even fill the ion flux HD plates with about 10 microliters of buffer, put those into the acoustic echo dispenser, and get your DMSO stock compound dispensed directly into the ion flux plate that way. And again, dilute with ECS just before the assay, and you're good to go. So all of this has enabled us to really increase the throughput and make life easier for screening on the ion flux. So in summary, we found the ion flux to be very precise. It has very fast liquid exchanges, and you get very quick responses from your ion channels. The complex liquid application protocols enable you to really normalize your data very well to internal controls, which is much more EFIS-like than perhaps traditional HDS platforms might allow. We've been running both ligand-gated and voltage-gated channels. I think in our view the ligand-gated assays are very, very good. And because of the standard plate format, it's very easy to integrate into screening processes. So with that, I will finish, and I will try and...
I'll take it back from you, Juha. So very nice presentation, Nice data. I have to say that the IT plots are really informative, even in terms of kinetics. You can really see a lot in them. They take a little while to get used to looking at, but once you get used to it, there's really a lot of information in there.
Absolutely, and I think that's the kind of direction that the world is heading to. Not to overuse the term that's already bandied about, I think big data is the way things are going, and if it's possible to analyze the wash-off kinetics of compounds, for instance, in addition to just the percent inhibition, then that is really very useful.
Yep, very nice. So in terms of questions, we're going to hold off on the questions until the end of the webinar. We'll be taking questions by the Q&A panel, and we will be compiling them. What we don't answer, we will answer by email. Adam, are you ready to go here for your presentation? Adam and Jim Harrington?
Yeah, just give us 1 second.
Okay. Sorry for the dead screen here. We'll have a presentation up here in a moment. Let me know when you're ready, Adam.
Okay, you can hand us the ball.
Okay. Okay, you've got it, Adam. I'll let you know when you, after you share your application, when I can see your slides. Okay, I can see them.
Okay, Jim's going to take the first part, and I'm going to take the second half.
Okay, great. Whenever you're ready, Jim.
All right, so today Adam and I would like to tell you a little bit about the electrophysiological approaches that we've taken to characterizing positive allosteric modulators of the alpha-7 nicotinic receptor. I'll cover a brief overview of the biophysical properties of the alpha-7 receptor and give you a brief overview of some of the signatures of these positive allosteric modulators as measured by manual electrophysiology. And then I'll turn it over to Adam, and he's going to show you how we've implemented a high-throughput automated electrophysiology assay on the Ironworks Barracuda that allows us to capture many of the features of these molecules. So just as a brief overview, nicotinic acetylcholine receptors are composed of 5 subunits, and each subunit has 4 transmembrane segments. We now appreciate that there's considerable diversity within this class of receptors, owing to the numerous subtypes as well as combinations. And we're going to focus on alpha-7. It's a major receptor in the mammalian central nervous system. And based on studies of the muscle acetylcholine receptor, we expect that the binding site for acetylcholine for alpha-7 exists at the subunit interfaces, and since this is a homopentamer channel, there's the potential for 5 equivalent binding sites for acetylcholine in this receptor. So there's been considerable discovery efforts targeting alpha-7 due to the potential utility of drugs targeting this receptor for the treatment of cognitive impairment associated with schizophrenia, as well as for the symptomatic treatment of Alzheimer's disease. And there are 2 basic approaches that have been taken. One is to develop agonists, either full or partial agonists at the receptor, and the other is to develop positive allosteric modulators, or PAMs, of the receptor. And these 2 types of molecules have very different properties. Agonists bind to the orthosteric acetylcholine site in the extracellular domain, whereas positive allosteric modulators bind to less defined, and probably there are numerous allosteric sites on alpha-7. Agonists directly activate the channel, whereas positive allosteric modulators have no effect in the absence of the endogenous ligand acetylcholine. When agonists directly activate the channel, this leads to essentially with chronic systemic administration of a partial agonist, for example, this really lacks the spatial and temporal resolution given by normal release of acetylcholine, and this can lead to desensitization with overstimulation, and can manifest itself as in a couple ways. One is with a inverted U-dose response curve in behavioral assays, as well as tachyphylaxis upon repeated dosing. Whereas positive allosteric modulators have a very different profile. They actually facilitate the phasic receptor modulation by the endogenous agonist and maintain those temporal and spatial patterns. And these molecules often have a very strikingly different effect on desensitization. They can slow desensitization, leading to prolonged opening and even reactivation of previously desensitized receptors. Partial agonists have progressed to clinical testing, whereas positive allosteric modulators to this point have not been reported to be tested in the clinic. So on this slide, what I'd like to do is just give you a brief overview of the basic conformational states of the receptor. Those are shown at the top. The 3 states are the resting or closed state, the excited or open state, and the desensitized state. And in the middle shows the transitions between these states. When the receptor binds acetylcholine, the receptor activates, opens briefly, and when acetylcholine levels fall in the synaptic cleft, this leads to dissociation of acetylcholine and closing of the receptor by a process termed deactivation. Now, in the continued presence of acetylcholine, the channel can adopt a desensitized conformation. This is a closed conformation with high affinity for the agonist. However, this desensitized state is not complete, so there periodically will be a small fraction of channels that will reopen by the process of reactivation. So this slide illustrates some of the basic electrophysiological properties of these various classes of molecules targeting alpha-7. Application of a full agonist, shown on the left, results in a rapid rising current as the channels open, followed by a rapid decay as channels desensitize. Currents produced by a partial agonist, shown in the middle, have similar kinetics, although they are smaller even at maximal concentrations of the partial agonist. Application of PAM, of a positive allosteric modulator, has no effect when applied by itself, but dramatically augments the currents induced by a submaximal concentration of acetylcholine. In this case, it's an EC20 concentration of acetylcholine. And in fact, the current amplitudes can exceed those produced by even a maximal concentration of acetylcholine alone. And the kinetics of the currents can be dramatically altered. And the challenge for a discovery program on this target is to implement electrophysiological assays with significant or sufficient kinetic resolution to measure these changes in channel kinetics, and with the throughput to drive an active medicinal chemistry program. So on the next few slides, I'd like to show you some of the manual electrophysiology data from this assay. So the basic outline is shown here. The human alpha-7 receptor is stably expressed in HEK cells, and currents are recorded by a Dynaflow resolved microfluidic system, which achieves rapid solution exchange. So the traces shown in red are essentially repetitive applications of a submaximal concentration of acetylcholine in 100 micromolar without the presence of a positive allosteric modulator. Then a test compound would be added by itself for 1 minute, followed by a repeat of this series where there's an application of acetylcholine in the continued presence of the PAM. And we chose 100 micromolar because that's an EC20 concentration as shown on the bottom right. And we would analyze 11 seconds of data. So the peak in area efficacy would be measured by essentially normalizing the responses to the maximal concentration of acetylcholine, and this would provide us a fold change in both peak as well as area. We would also measure EC50s based on both the peak and area measurements. On this next slide, give you an example of a further metric that we employed to study deactivation. And the currents were integrated over this 11-second period. First, the area during the acetylcholine on phase, or the activation area, the 1 second when acetylcholine is applied, that area would be calculated, and then also the 10 seconds of the time when acetylcholine is off, the deactivation area. And the metric that we used was the deactivation number. This is a fraction of the fraction of the total area that is made up from the deactivation area, and it provides a measure of how much a positive allosteric modulator slows the deactivation process. This slide shows some examples of different types of PAMs as classified in the literature. A type 1 PAM just increases the peak current. And so what you see on the top of this slide in blue are the control responses to 100 micromolar acetylcholine. And then superimposed in green are in the presence of a type 1 PAM. And you can see that the type 1 PAM just increases the peak current, has no effect on desensitization or deactivation. In contrast, type 2 PAMs slow desensitization. And we adopted a further subclassification of type 2s into A and B. Note that the type 2A PAM slows desensitization but has a modest effect on deactivation, whereas the type 2B PAM has both a profound effect on desensitization, so there's a large increase during the activation phase, as well as a dramatic slowing of deactivation. So on these next few slides, what I'd like to do is show you some examples of compounds that fit into these various bins. Each example has the same series of plots. On the upper left, a plot of the normalized current response by measuring the peak current versus compound concentration at an EC20 concentration of acetylcholine, and that's why the curves plateau at a non-zero value at low concentrations of compound. And then on the upper right is a plot of the fraction of the total area measurement coming from the activation phase in green or the deactivation phase in orange. And on the bottom are the raw traces. In addition, it's reported the area fold change. So this is on this slide, this is an example of a type 1 PAM. You can see that it doesn't change the channel kinetics, just increases the peak. Its deactivation number is 0, and it has a modest effect on the area. There's a fold change of about 1.6 fold. On this next slide, it shows an example of a type 2A PAM. This compound slows deactivation. You can see that on the bottom raw current traces, but doesn't change deactivation very much. Hence, the deactivation number is still quite small at 0.08. It has a significant effect on the area fold change. It increases the area 40-fold due to the slowing of desensitization during the acetylcholine on period. This next slide shows an example of a type 2B compound. We arbitrarily set the criteria between type 2A and type 2B at a deactivation number of 0.5. So this compound has a deactivation number of 0.67. And you can see that looking at the bottom traces, that there, in addition to a slowing of desensitization, there's also a significant slowing of deactivation, and that the area fold change for this compound is significant too. It's at 52-fold. On the next slide shows the example of an extreme type 2B PAM. In this case, the deactivation number is 0.9. And the reason being because the most of the area coming from the integrated current is due to this dramatic slowing of deactivation, as you can see on the bottom, and hence the fold change is extreme at 900-fold increase in the total area under the curve when we're measuring the integrated currents. So as you might imagine, these are just some examples of the types of profiles one can observe. And really, in practice, when one studies many compounds, there's really a continuum of kinetic signatures that even within a single structural class. And really, the challenge for supporting a medicinal chemistry effort on such a target is to find higher throughput electrophysiological assays that can capture some of these kinetic details while reliably reporting potency estimates with cycle times to rapidly drive compound design. And so for this reason, we made a concerted effort to develop an assay for type 2 PAMs on the Ironworks Barracuda platform. And I'll now turn it over to Adam, who led this effort.
Okay, thank you, Jim. So I think that the manual patch clamp work on this program was absolutely elegant. Definitely developing some of these advanced metrics were really key in drilling into the pharmacology of what we were seeing, so it bears mentioning that a lot of these compounds that Jim's showing are stuff from our deck. So it's novel starting points for alpha-7. So I'm going to gear shift a little bit and talk about sort of the pragmatics of the hits to leads approach around alpha-7, but a lot of this sort of generalizes to more broadly to the ion channel space. So there's a lot of slides like this out here. This is just sort of my version of it. So this is sort of of course we have the hit identification phase of the high throughput screen, which generates hits, which if they're lucky, they sort of wriggle upstream if they generate the appropriate selectivity and physicochemical parameters. They become leads and starting points for our SAR efforts. Typically, in drug discovery world, the HTS toolbox is in one sector, and these are typically optical assays. And in the case of an ion channel, the secondary hit assessment, all the biophysical characterization and selectivity is done with electrophysiology. We love electrophysiology in these programs. It's probably the most information-rich functional assay out there. It was a translational assay before that was a thing, right? But under the sort of constant specter of time pressure to get these things done in a meaningful amount of time, as Jim mentioned, it's very useful to find ways to sort of narrow this gap between the primary hit identification and the secondary hit assessment, which are done sort of with different assay modalities. And that's sort of where the challenges lie in bringing these optimized leads forward at a meaningful amount of time. So specifically with alpha-7 PAMs, obviously, as Jim mentioned and covered very well, there's a lot of interesting biophysics with the molecules that have been reported on in the last 10 years or so. Here's one that apparently can, and with kind of interesting kinetics, reverse orthosteric agonist-mediated desensitization. There's an evolving book of work on these types of compounds, and the feeling is that these different types of mechanisms, type 1 versus 2, and now 2A versus 2B, could be the key to differentiation in this space, right? So we want to differentiate first preclinically and then, of course, ultimately in man, a partial agonist from a PAM, and we also like to find a way to differentiate among the different types of PAMs, in models and in patients. So our program here at BMS hit on this sort of granular electrophysiology workup of the leads as a really big determinant of success for the program, and that set us on the path to find ways to move this along in a meaningful amount of time, because we have typically in ion channels a lot of hits and can create a bottleneck situation around hit assessment, and that's what we wanted to try to avoid here. The approach that was taken was our first foray in leads at BMS into population patch clamp. So for that we use the Ironworks Barracuda. A lot has been said about this. Obviously, it's population recording that gives you the plate and assay architecture of a flipper assay, but with the fidelity to the native functioning of the molecular motor of the channel of a patch clamp assay. So basically, it's a win-win in all senses. And this assay format makes the handshake with optical hit identification assays pretty seamless, and it gives us a throughput to get through a lot of hits and get to our interesting leads for these secondary, more granular manual patch clamp assessments all the much faster. So it's a very profitable innovation for us. This slide looks awfully familiar to the one that Jim showed earlier, so I won't spend a whole lot of time on it. It's kind of a worst case, best case scenario, sort of tale of two approaches. So on the left here in funnel, we have, and this is sort of, again, this could be alpha-7 PAM, it could be any ion channel program. We have generally these optical HTS campaigns, depending on the number of compounds you screened. It's very common to have, hits in the tens of thousands. There on the left, if you don't have an electrophysiology assay positioned fairly far up front in your triage, and we tried to push ours as far up as we could, you end up getting creative with other types of ways to sort of bend down to find the really interesting compounds at the end. And if you look sort of there at the bottom, you can find yourself in a situation where you're confronted with a huge onslaught of compounds for your typically, generally, relatively constrained manual patch clamp resource. And it's difficult in these ion channel programs to bring leads forward at a meaningful amount of time faced with these constraints. Whereas when you have the option of using population patch clamp, you can bin compounds right out of the screen, right after the CRC step very early, from thousands to hundreds, and get down to very, very manageable numbers of compounds for your detailed, more granular secondary biophysical analysis, and also for confirmation step in the SAR process. And we do that, we'll talk a little bit more about this later, but we do this by leveraging the capacity of the Barracuda, the flexibility, and also our ability to reduce data in our offline informatics environment and create a large amount of ready-to-use analyzed data in a really short amount of time, which is something that often you can't do with manual patch clamp as well, because we just don't have the plate format flexibility to bring that off. So, for the Barracuda crowd out there, I thought I would just detail the assay a little bit. This assay sort of, as they do, kind of evolved over time, but what we ultimately used to drive the program over the roughly 9 months that we were pushing secondary hit assessment and SAR support at the full discovery phase was a PPC assay. We brought the PAM in at 3x, so it was there was a dilution step there. We did a slow mix and we waited 5 minutes. Then we brought in another slug at 1x, so this was a push-through with the PAM plus a rough EC20 of the orthosteric agonist. And this was done as fast as the cells would tolerate, which ended up being about 10 microliters per second. I think the instrument goes a little faster than that, but you start blowing off cells if you do that. You can sort of see down there on the bottom right, here's some of the raw biology. It's just kind of a teaser. This is stuff from our deck. So as you can see, we saw a lot of different, sort of a range of potency and efficacy. This was a flipperized assay, which is sort of, by which I mean that the compounds are dosed out across the plate. We're not doing in-well titrations, because we did have, with our alpha-7 RIC3 cell line, we had a real good reagent, which was heavily clonal, very homogeneous population of alpha-7 expression, and that's obviously the big trick in designing cells for population patch clamp, is you need a high degree of clonality or it's not going to go anywhere. So this was our workhorse hit assessment assay, and also our SAR pre-screen, and then we passed compounds over to Jim's lab for the type of granular biophysical assessment that you just saw, and that worked really well. It was a good approach for us. Allowed us to get away from the bottleneck scenario that I was describing, which does sometimes attend ion channel programs, and we found innovative ways to extract a fair amount of information content from both the manual patch clamp assay and from the Barracuda, and I'll just sort of detail some of that. So here on the top left, I've got some red meat, so to speak. There's a number of compounds titrated in that plate. I generally don't like to just show screenshots of plate maps, but I think it underscores the capacity that we had with this assay. So the 16 CRCs there, obviously a range of potency, efficacy, and apparent desensitization kinetics, but we could put 32 in a plate, and we often did. So at full discovery, we were putting out roughly 150 compounds a month, and we were able to handle that in one afternoon a week. So in a leads group, you sort of have, it's kind of a data factory. So you have things sort of, the assays run on a revolving schedule, and we don't have all day, every day to run these types of assays, and that's, to some degree, prior to the advent of the real high throughput patch clamp robots, that's sort of something that we had to contend with. With the Barracuda, you can plug it into a Tuesday afternoon spot and get all your data, right? So we get the raw biology, reduce it in the offline informatics environment. Here to see the heat map, and then it's fitted and extracted by the chemist for his or her use and pushing the SAR process forward. So this is what our workhorse assay looked like, and it was sort of down to us to try to find ways to mine as much information out of this as we possibly could. So a couple of caveats with these assays, sort of before I detail this slide. I mentioned I was using a rough EC20 of acetylcholine, because I don't know what the acetylcholine CRC looks like in the Barracuda. The acetylcholine response at alpha-7 is too fast to see in the population readers generally. So the way that's one of the reasons why I always say you sort of need a blend of instruments for doing these ion channel programs. It's absolutely essential to have the ability to go into manual patch clamp with ultra-rapid solution exchange and do that type of stuff. The way we got to our assay was basically holding a PAM, basically titrating acetylcholine versus a fixed concentration of a PAM, and sort of a reverse shield type of analysis, and working to a place where we identified the acetylcholine concentration that gave us the most potent PAMs that weren't running into a big ceiling effect with efficacy, because all the sort of normal constraints around designing positive modulator assays that you see in optical assays also apply here. So that's the way this assay was designed. We ended up with about 300 micromolar acetylcholine on board. That jives pretty well with the rough EC30 in manual patch clamp. So we feel like and as you can see here, the pharmacology across the two platforms was very well aligned. We were using this as a predictor and as a triage tool, and in that sense it worked really, really well. Sort of on the bottom left there, we did do an optical HTS. It was sort of done in a couple of phases, and then early on we were trying some different approaches with the optical assay to sort of tease out a little bit more information. Ultimately, that helped us in a sense that it sort of focused us on some of these more biophysically novel type 2 PAMs that it really didn't get us over the hump and sort of hit assessment. It was really not until we brought in the Barracuda that we were able to really do that. And you can kind of see the other issue with the relying on the flipper for SAR was it was kind of giving us a two-way miss. Were some compounds that would significantly overstate the potency relative to patch clamp, and some compounds that would almost totally miss it. Whereas in the bottom right correlation plot there, you can see that the Barracuda and the Dynaflow manual patch clamp assay lined up really well. And this is just a smattering of our tool compounds, some stuff from the literature. But as the program went on, we were able to build these plots out pretty significantly, and always we saw a very good predictive ability for the Barracuda, which is key, right? Like I mentioned, the Dynaflow assay is an incredibly high information content, but it's also labor intensive and not terribly high throughput. So the role of the Barracuda in this was directing that really, really precious manual electrophysiology resource in the right way, because we've all seen programs where that gets directed in the wrong way, and then there's negative consequences downstream in terms of just progressing things in a meaningful amount of time. So here on slide 20, this is some more raw data, and I just sort of highlight it. You can see obviously that there are a range of potencies here. Some of this is some of our early chemotypes. There's a couple of literature compounds thrown in here as well. I apologize there's no scale bar, but the little blocks are about a second. It gives you a sense, and the larger response there are about 2 nanoamps. So we obviously have a range of potency and of efficacy and also of apparent desensitization kinetics. And upon seeing this, it pushed us a little bit to try to come up with some type of an advanced metric to pull a little bit more information out of this. Now, bear in mind, in the Dynaflow, we can switch acetylcholine on and off in the presence of the PAM and record all the while. The Barracuda, this is a plate pharmacology assay. We can't do that. When the acetylcholine is on board, it's on board. It doesn't wash out in the midst of a response. So we weren't able to make a direct type 2A versus 2B differentiation, right? Because the key there is to be able to remove acetylcholine and see if the channel is being locked into this slow deactivating state, or whether the PAM response basically attends the acetylcholine response in terms of time. But thinking about it, we were able to come up with another sort of SAR vector around this that, again, let us do a little bit of binning for the secondary SAR, or for the secondary biophysical workups in Jim's lab, and that was just to basically use MATLAB to do a relatively straightforward area under the curve transformation. And this was done by our colleague John Grave in the genetically defined diseases department. He's very good at writing scripts in MATLAB. So we came up with an automated MATLAB area under the curve transformation script, whereby we were able to pull that out for all of our compounds, basically in parallel timing with the extraction of the data for potency and efficacy. And this let us do a bunch of interesting stuff. So, as Jim mentioned, the deactivation number is the product of dividing deactivation area by total area. That's an advanced metric that Jim's lab devised to sort of drill a little bit more into the different types of type 2 PAMs that we were seeing, because this is, a lot of the stuff that we got out of the deck was relatively diverse structurally, and it was definitely diverse pharmacologically. So we needed some tools to kind of bin this out, because the goal of the program, and we didn't, because the program is no longer in play, we didn't achieve this, but the goal of the program was to identify parallel SAR work streams around these different types of PAMs and then see if they could differentiate in animal models, both versus themselves and versus a partial agonist. So Jim mentioned that the deactivation number, deactivation area divided by total area, below a deactivation number of 0.5, typically these are type 2A PAMs, and above a deactivation number of 0.5, typically these are type 2B PAMs. So looking at this graph here on the middle left, you can see when we plot the Barracuda data versus the Dynaflow deactivation, it's a very reliable correlation below 0.5 in the sense that if we know that we're in a type 2A chemical space, the Barracuda area data can be relied upon to track very closely with the Dynaflow deactivation number, whereas in the case of 2B BAMs, that sort of falls apart, right? And then the reason for that, as I mentioned, is we're unable to make a determination in the absence of acetylcholine in the Barracuda. That's the limitation of the plate pharmacology setup. Correspondingly, here on the top right, in settings where acetylcholine and the PAM are both on board, the activation area in the Barracuda, or in the Dynaflow activation area and the total area in the Barracuda, the correlation of responses for tool compounds, it's pretty good. Not great, but it's more than good enough to sort of guide us in binning compounds and starting to understand this, whereas when you look at Dynaflow deactivation versus the Barracuda area, because we're not able to make a determination in the absence of acetylcholine on the Barracuda, it starts to, it follows that the correlation starts to fall apart, and that's borne out by the data. So this was towards sort of some advanced metrics for allowing us to effectively triage compounds for what goes where, and it was also towards pushing that Barracuda further and further and further up the funnel in terms of SAR drivers. As our understanding of what we wanted in a compound evolved, we felt like we were moving towards being able to leverage that type of information from the population patch assay, which was a fun process and very instructive. The Barracuda is a very versatile platform, and we found that our experience in doing alpha-7 modulators was just very, very good in the sense that we got a robust assay with a lot of throughput. We were blessed, of course, with a good cell line, and it allows us to move from the hit assessment phase into SAR, certainly fast for an ion channel, and almost fast for any type of program, and get into the medicinal chemistry process and make some pretty good strides in a relatively short amount of time. So I'm not sure where we are for time, but I'll just wrap it up here, summarize Jim and I's presentations. Basically, the manual patch clamp gives us a robust, manual patch clamp is a robust platform for measurement of alpha-7 PAM kinetics. You can do these very granular workups, as Jim very elegantly showed. And the second step in the 1-2 punch was the Ironworks Barracuda, which provides rapid hit assessment and high throughput SAR on a range of metrics. So I think we're maybe going to take questions at the end.
Yep, Adam. So
yeah, before I go, it's always a team effort, of course. I'd just like to acknowledge everybody that worked on this program. It was a fun effort, and I think we made some good strides in sort of our understanding of the modulators in this space, as well as some of the technology that it takes to get that done. All right.
Okay, yeah, Adam and Jim, I want to thank you very much. As you said, very elegant data in both manual and on the Barracuda. And I'm actually, not being a PAM expert, the kinetic effects of the PAMs are so dramatic. I can see why it's such a hot topic in drug discovery. As you mentioned, we will do the questions at the end. We have one more talk from Ali at Fluxion, and so I'll go ahead and I'll pass it over to him now, and I have to unmute him as well. So, Ali, it's coming your way. And I just unmuted you, so,
Okay, can you hear me now?
yep, you sound just fine.
Okay.
I'll mute the other participants, and then we'll unmute everybody when we're time for questions. So, Ali, whenever you're ready. Okay. I can see your slides.
You can see my slides. Okay, perfect.
Yep.
Okay. So my name is Ali Yahya, and I'm the director of iFlux product development at Fluxion Biosciences. After these two great talks before me, I would like to expand a bit about the capabilities of the iFlux system for enhancing PAM assays development. First, a bit of background about Fluxion Biosciences. The company was founded in 2005, and our headquarters are in South San Francisco in California. Being a small company, we have many distributors worldwide, and the company won 5 NIH grants and 2-time R&D 100 awards for BioFlux in 2008 and IronFlux in 2010. We have 3 main product lines, all based on microfluidics technology. BioFlux, used for live cell imaging under flow, IronFlux, our offering to the automated patch clamp arena, and IsoFlux, a microfluidics-based CTC isolation system. Now, the IronFlux system is built by Fluxion Biosciences, but distributed and supported by Molecular Devices. The IronFlux is, by simple terms, an automated patch clamp system based on a unique in-plate liquid handling or solution movement strategy. The system does not require a complicated liquid handler to move solutions, and hence has a footprint of a simple plate reader. It comes in 2 main flavors: IronFlux 16 with 16 amplifiers, which is a great entry point to automated patch clamping, and an IronFlux HD with 64 amplifiers, a great contender and step towards the medium EFIS high throughput market. The lack of liquid handler allows both IronFlux 16 and HD to be quite competitive price-wise and great value for investment. Okay. However, effect on system pricing aside, this lack of dedicated liquid handling is actually the end result of the IronFlux unique ability to move solutions inside of the recording plate itself. The machine uses standard SPS plate formats, 96 wells for IF16 and 384 wells for IronFlux HD. But the difference is that the bottom of the plate has been replaced with an intricate laminar flow system, providing an integrated cell and compound delivery as well as recording. Looking at the bottom of the IronFlux plate in more detail, the plate is divided into patterns, as you can see here. Zooming into one of these patterns and then flipping it to look at it from the top, the pattern is composed of 12 wells, 2 independent recording wells connected to separate amplifiers. These recording wells, also called traps, would contain the intracellular solution, an inlet well where the cells in extracellular solution are introduced, an out or waste well, and 8 different wells where different compounds and solutions can be added. We have 32 experimental patterns per IronFlux HD plate and 8 patterns per IronFlux 16 plate. The design of the plate and the intra-movement of the solutions allow for independent compound conduits, hence no mixing, no cross-contamination, no pipetting delays, and no electrode-compound contact. The microfluidics design was featured on the cover of Assay for August 2012. We like to show this all the time. We also had an article in that issue, but nevertheless, the cover is beautiful on its own, as you can see. Okay, now looking closely at the mechanism of action. Once plate is filled, it is introduced into the machine. The start of the assay begins with priming of the microfluidics. After that, the cells with extracellular solution start flowing through the main chamber until trapping occurs, and seals are formed. And then afterwards, extrasuction opens the cells and recording begins in whole cell patch clamp mode. So a typical experiment would involve opening of the compound pneumatic valves, allowing the release of the compounds into the main channel, flowing continuously towards the cells. The independent control of these compound inlets provides fast and successive compound additions, as well as continuous compound washing in between the compound additions. Consider that these wash steps are not really steps, but more like washing gaps in between the compound release from their inlets. So this will become clearer later on. It is important to note that the reliance on pneumatics instead of physical robotic liquid movement provides the parallel release of all compounds at the exact time. So the whole plate is done all at the same time. Hence, one experiment or 32 experiments will take exactly the same time to complete, allowing the influx assays to complete larger experiments very quickly. Okay. Now, as mentioned, the influx plates record from cells in whole cell patch clamp mode. Two types of plates exist. Ensemble plate with 20 cells per recording. Those are the most common ones. And single cell plates, which a lot of people use also for different needs. Ensemble plates are designed for high quantity in mind, where currents from all cells are added, not averaged. And single cell recordings are meant for highest quality, where cells are patched and sealed with gigaohm seals. Let's look back at the solution movement and its control within the plate, since that's the most important aspect for PAM studies. Similar to what Juha presented before, the influx software provides an extensive graphical control over the delivery of compounds and solutions. To start, the start and end time of the release of a specific compound is simply drawn in the software. Separating the steps would mean washing of the cells. The longer or shorter the distance between the steps, the longer or shorter the wash step. In fact, we can get the steps so close to each other that no washing occurs. Since this is a continuous flow system, washing is continuously going. You just need to factor it into your assay. Here we step from one compound to another instantly. Again, all that is happening in parallel within the plate. Although these steps are of exact size here, you have complete control over their lengths and can even superimpose them, just like what Juha showed. This ability shows off the true capability of the system's fluidics. For example, we can have two compounds flowing at the same time, here from well 2 and well 3, with the cells only seeing the compounds flowing from well closest to them, here well 3. Then we have an interruption in the flow of well 3. The compound from well 2 would fall on the cells nearly instantaneously. This flexibility in compound displacement provides extensive capacity in development of difficult ligand-gated assays such as PAM assays and double agonist assays. I'd like to show you a few examples of such assays using GABA receptors modulation as targets. To start, we have used hex cells expressing GABA A receptors, here alpha-1, beta-3, gamma-2, millipore urafin precision cells. The plot shows serial sweeps from the same recording while superimposed demonstrating the agonist activation of the receptor. Now these are serial sweeps, so that is like one sweep after the other in the same well. When compiled together, a dose-response curve can be plotted showing an EC50 of 3.32 micromolar, which is consistent with published values. This is N equal 40. Now the receptor is sensitive, of course, to a number of agonists, including GABA, muscimol, and isobutyrin, and even ethanol. Here is the response to 3 micromolar muscimol and 3 micromolar GABA and 3 micromolar isobutyrin, showing the different potency of the 3 different compounds. Now here you can see actually that potency for muscimol was higher than GABA, higher than isobutyrin. Again, the EC50 values were consistent with published results. Now, just to establish our baseline with regard to our assays, it is important to note that both positive allosteric modulator and the agonist are needed to initiate a response. An agonist alone can open the channel with the PAM increasing the effect of the agonist. However, if the PAM is added by itself, no response is seen. Hence, to study PAM effects on receptors, the baseline effect of the agonist need to be consistent and repeatable, in addition to the already mentioned highly flexible and speedy liquid control. So we really need to establish the repeatability of the baseline response. So here, what we're looking at, we're looking at the repeatability of the baseline response with regard to GABA. The rapid control of solutions as well as the continuous flow of compounds provide the influx with consistent response to the same agonist addition. So although we may have differences in the total value of the current, the sweep-to-sweep variability is basically minimal. Here we see 2 recordings wells receiving 10 micromolar GABA. Notice the currents are not of the same size since they come from different wells. However, sequentially, variation from sweep to sweep is nearly minimal. This can be observed more clearly with this IT plot. So just to explain about these IT plots, which are actually seen live when you're actually recording from the influx. This is a plot of values seen by placing cursors in the software at specific location in the sweep run. So within these sweeps, two cursors are placed, one before the addition of GABA and one after it. The cursors measure mean current and the values are subtracted. When GABA is added, a response is measured and a negative point is recorded. When watch steps are followed, no response is seen and a zero value is recorded. So this repeatability and consistency of response is crucial to establishing a baseline for the study of PAM effects, since sweep-to-sweep response to GABA would become our control. Okay. So once we establish repeatability and consistency in basal GABA response, we can look at the effectiveness of the successive serial additions in PAM assays. In this experiment, we first had 1 micromolar GABA added, establishing a baseline response to agonist. Next, 0.05 micromolar diazepam is added and incubated for 1 minute. As expected, no current is observed. However, when diazepam and GABA are co-applied, a slight increase in the current is observed. Next, another incubation phase is performed, but now with 0.15 micromolar diazepam, which also shows no evoked current. Finally, 1 micromolar GABA with the 0.15 micromolar diazepam would result in current which is substantially larger. So all of this, again, is serial, is one after the other, with no watch steps in between. It just goes on. Here we have the successive superimposed sweeps from the same recording well, showing the effect of increasing diazepam concentration on GABA currents. The baseline is established again with 1 micromolar GABA, followed by 0.05 to 20 micromolar of diazepam. We also tried PAM of the same benzodiazepine family, like triazolam. Note that the higher potency of triazolam as compared to diazepam, and the distinctive effects on receptor potentiation. Now, the EC50 of diazepam, zolpidem, and triazolam, and zolpidem is actually marketed as Ambien. I can see the commercials for it most of the time on TV, and is outside of the benzodiazepine family. Experimental values were 425 nanomolar, 84.6 nanomolar, and 12.1 nanomolar, which are in agreement with literature. Note the increase in potency of triazolam followed by zolpidem and diazepam. Now, the pneumatic control and the in-plate solution movement allows the influx to have the feasibility for automated assays that are challenging on systems which are mostly dependent upon liquid handlers to move solutions. Looking back at the example compound addition map from before and considering that the activated GABA A receptor current shows a fast activation followed by a slow deactivation, it is possible to sequentially increase the agonist dose faster than the rate of deactivation, effectively increasing the activation of the current in a staircase effect. If all timed correctly, a current staircase would be seen with the same sweep in response to increase in agonist concentration. An example of this, the staircase activation is shown here. The receptor was activated with 1 micromolar GABA, followed by 3 micromolar GABA, and then 10 micromolar. The compounds are applied for 2 seconds at each concentration. Here we have response for 2 wells. An interesting part of this assay is the ability to obtain a quick EC50 from a single sweep. Again, this is not calculated current versus time achieved by obtaining multiple sweeps. This is a staircase increase in 1 sweep. Now, to show the potentiation effect of PAM, a similar experiment is conducted where a constant application of modulator is added and compared to control. Here we have the same staircase GABA activation with and without the application of 3 micromolar diazepam, clearly showing the expected allosteric shift in current magnitude. Now, of course, this is similar to what Juha has talked about before. It's relating, so these rapid liquid displacement assays can also, of course, be seen for the open channel modulation assays. And here looking at an open channel assay similar to what Juha showed. In this example screenshot, cells were perfused in 1 micromolar GABA for 4 seconds, then switched, that's where the arrow is, to a GABA diazepam mixture solution for 8 seconds. Different traces in the graph are recordings from 16 different cells ensemble. The effect above shows the open channel potentiation of the receptor, and of course the assay can be expanded to other PAM targets. Another example, here is an overlay of DMSO control in red, in green showing GABA application for 8 seconds, in blue GABA for 4 seconds, then GABA plus diazepam for 8 seconds, and in orange GABA for 4 seconds followed by GABA with a blocker, here picrotoxin for 8 seconds. All of the above experiments were done within the same plates and at the same time, again with all of the steps within the same sweeps. Okay, these assays can be used to investigate other targets, of course. So now that we have established that we can do all of these fluidics and all of this movement of solutions, what about more difficult targets? For example, NMDA double agonist. So this is actually, just going to show you this slide, which is very similar experiment. Here, a cumulative concentration response with NMDA. Shown are 16 ensemble recordings from a single plate. Glutamate concentration is increased with basal concentration of 30 micromolar glycine. As glutamate is increased, the response is increased. It is important to note that NMDA experiments do benefit from enhanced fluidics of the influx system and the continuous flow. Considering that these experiments are quite difficult in stagnant solutions due to the open channel toxicity of NMDA receptors, which is induced by local production of glycine and glutamate from neighboring cells. So as summary, the influx system, equipped with its unique liquid exchange and continuous flow, provides an excellent platform for complex PAM assays. The ability to provide a consecutive and fast switch from one compound to another or to serially increase compound concentrations with no wash steps or liquid stacking problems gives the platform the unique edge and speed a complex PAM qualification assay would require. Thank you. Jim? You can take it back.
Thank you, Ali. Thanks for walking us through the influx instrument and its capabilities. I have to say we really enjoy working with you and with Fluxion to bring customers the influx technology.
Same here.
With that, we're going to go ahead and have a short question and answer period. We've been taking questions through the Q&A tab. What I'm going to do at this time is unmute the participants and also Shin, and then he's going to go ahead and ask some questions. And I hope that technically I do this correctly. So, Shin, you should be able to hear me now. Is that correct?
I can hear you now. Can you hear me?
Yeah, I can hear you just fine.
Excellent.
So I'm going to give my. You can go ahead and just start asking away. I'm going to go ahead and put some slides up here while you're working.
Okay, sounds good. So I have a number of questions for each of the speakers. Actually, very nice presentation. So maybe I'll start with Juha. Juha, are you on the line?
Yep, I'm here.
Okay. Okay, excellent. So there are a couple of questions surrounding how the influx systems fit into the drug screening process in Nucentis. The first one would be regarding the data analysis. How do you use the influx system, or how do you process the data collected from the influx systems for your need?
So for those of you who haven't worked with the software before, and you know, Ali can also comment on this, the influx software is primarily sort of intended for a very quick selection of data and then exporting that. So the way we analyze the data is as I presented with the either with the gene data analyzer package, which is very useful because you can load multiple plates worth of data into that, and then apply standard measurement and calculation cursors, and then export a file with your numbers into another database. The other way that we're doing things is that we're essentially exporting all of the data into the IDBS Biobook software, and we've got some template spreadsheets in there where we can apply QC for things like seal resistance and minimum current amplitudes, and then those Biobook spreadsheets calculate our either percent inhibition or percent enhancement, and then we're able to directly publish our data into databases from there. So those are the primary two ways that we're analyzing the data.
Excellent, thank you. The next question, since you mentioned in Nucentis you have a number of automated electrophysiology instruments, and so the question is about how the influx HT in terms of the throughput compare with the other systems you have.
So I would consider it a medium throughput system within the world of automated EFIS. So as you mentioned, the Barracuda is at the top end of the throughput, but as examples of what we've done in real life, we are able to easily support library chemistry, where the chemist submits compounds for testing for us, ranging some tens of compounds to a couple of hundred compounds per library. and we were able to get those done in a week. And these are the types of assays that I showed in my presentation. So it's concentration responses for that number of compounds. So, for instance, a couple of hundred concentration responses in a week. And that usually actually requires, 2.5 days, so it isn't even a full week.
Excellent. Thanks, Ilhan. There may still be questions coming in. So for now, maybe I'll just move on to Adam and Jim, and a number of questions about Barracuda and PAM assay for the Alpha 7. Are you guys online?
Hi.
Okay, excellent. So, so the first question is about if you can use the Barracuda system to measure the shift in the PAM potency with acetylcholine, like a shield type of analysis.
Yeah, that's possible. It's sort of it would be a modified shield type assay because, as I mentioned, we can't see it. The way you might normally think to do it would be to run your acetylcholine CRC in the presence of increasing concentrations of PAM, but since we can't see an acetylcholine CRC in that platform, at least not remotely consistently enough to run an assay around it, we would basically flip that and titrate the a CRC of the PAM versus increasing concentrations of the agonist. And in point of fact, we did that when we were designing the assay just to figure out what concentration of acetylcholine was optimal. So yes, you can do that with the PAMs, and if you had an agonist that was, like GABA or something that was slow, you definitely could do a conventional shield type of thing, no problem. Right. And something actually that Jim and I have been talking about doing a little bit of for sort of some of the wrap-up on the PAMs.
Okay, great, thanks. I think also the second question I think you touched upon in your talk is how do you measure or if you can measure the deactivation of the alpha 7 channel regarding the PAM effects?
Well, right, as Jim mentioned, there's a number of ways to look at acetylcholine independent effects on deactivation. The way that we took to sort of drill into that was generating the area under the curve metric, and then basically doing some post hoc correlations with manual patch clamp data to sort of understand how those two things match up.
Yeah, I mean, one other thing we thought about was trying to do sort of a double addition where we go in, and apply the PAM and then go back and then do a wash to try to get at some measurement of deactivation. It's conceivable that we could probably discriminate, deactivation for a 2A versus a 2B PAM, but it's unlikely that we're going to get really precise estimates of what the off rate of acetylcholine is under those conditions.
Understood. Okay. So let's move on to Ali. There are a couple questions for you, Ali. Can you hear me all right?
Sure. Yeah, I'm here.
Okay. So you describing in your talk the workflow of the ion systems, and question number one is about the software. Specifically, can you run multiple voltage protocols in sequence on the ion flux system?
Yes, actually, in the present software, which is available right now, the procedure was that you can actually run a voltage protocol, and then you can actually pause, change the voltage protocol, and then you can resume again. That's like a manual labor, but you could do it. However, with the new software, which is just released right now, you can actually put them in series. You can we call them multiple data acquisitions. You can put them in series, and then afterwards you can just tell the software, this is the first DA, this is second DA, this is third DA, and then you can save them directly into the actual script and then just run it directly like that. You can still pause and resume and change all of this still, but now it's a much more flexible. Plus also all of the compound additions that you have set up, if you have set them up in the different protocols, when you combine them like this, they all combine and you can visually see all of your additions. This is version 3.5, which we just released. I think it's being installed like as we speak.
I see. So this is like the sequencing key in the P-clamp software or something like that.
No, actually, with the P-clamp, we are actually building up a bridge for P-clamp. That's actually something else.
Okay.
So with regard to the analysis, so as you have mentioned, the software presently, what it does is that it actually acquires mostly, and it exports the data out. So it does have a bit of analysis into it, but there's a lot of room for improvement. So instead of actually building our own analysis package and things like this, one of the perks you can say of our collaboration with Molecular Devices is that we can actually build the bridge with P-clamp, and that's what we're doing right now with the software, which is in development. Software coming up later is going to allow you to acquire in the Enflux, press a button, and then P-clamp will see the data, and then you analyze in P-clamp.
Excellent. All right. I think that's a question so far we have for all the speakers. Thank you very much.
Okay, thank you, Shin, and thanks again to the speakers. What we're going to do is we'll leave the line open now for about 10 minutes or so. So if you have questions that you want to continue to ask us, just send them into the Q&A panel, and we will answer them by emails. I just wanted to say also at this time, we will be having another webinar in about a month or 6 weeks, so in mid-July or so probably. So I'd like you to tune in for that, and you can also see the recorded webinars on our website. And if you liked what you saw, you can also request a quote on our website. With that, we'll go ahead and leave the line open, and we'll mute everybody, and we'll be just taking your questions by the Q&A panel for the next 10 minutes or so. I want to thank you, and we hope to see you in July for our next webinar. Thank you very much.
Thank you.